Very good afternoon to all. Respected Vice Chancellor, respected Dean, School of Postgraduate Studies, respected Dean Agriculture, and the external speaker, Dr. B. Ramakrishnan, and my dear student friends and faculty which are online and offline, and students which are who are online and offline. So I am very happy to welcome you all for this lecture. And our speaker is eminent microbiologist working in Indian Agricultural Research Institute, and he has been working in microbiome of soil and plant. And in this juncture, I welcome you one and all for this meeting, and I request our respected dean to introduce the speaker. Thank you.
Good afternoon to everyone who have joined online and offline. So today we have gathered here for endowment lecture on microbiome innovations for the future of food and agriculture. So to deliver the endowment lecture, we have the very popular personality, Dr. B. Ramakrishnan, Principal Scientist from Indian Agricultural Research Institute, New Delhi. On behalf of ACNRI Madurai, I wholeheartedly welcome our guest speaker for this session. So in this context, so let me introduce our guest speaker for the benefit of participants who have joined online and offline. Dr. B. Ramakrishnan, Principal Scientist from IRI, earned his bachelor's in agriculture from Tamil Nadu Agriculture University, Coimbatore. So we are happy to have our alumnus here. So on winning the Indian Council of Agricultural Research Fellowship, he went to GP Pant University of Agriculture and Technology, Pantnagar, for his master's in microbiology. Dr. Ramakrishnan worked for his doctorate in microbiology from IRI in New Delhi. Immediately after his doctorate, he joined the Agricultural Research Service, that is ARS, of the Indian Council of Agricultural Research in 1992. His first assignment was at Central Rice Research Institute, National Rice Research Institute now, Cuttack, Odisha, from 1993 to 2010. Later, our guest speaker joined the Division of Microbiology at IRI, New Delhi in 2010. So because of his research, he has published many of his research articles in reputed journals. So his publications are available in Google Scholar. His current research interests are on microbe mediator nutrient cycling, microbiomes of soils and plants, and the role of microorganisms on health. Dr. Ramakrishnan is an expert member of the Ministry of New and Renewable Energy, Government of India, and the Fulbright Fellowship Committee of US Educational Foundation of India. Our guest speaker serves on the editorial boards of Current Science, then Frontiers in Microbiology, and Frontiers in Environmental Science. So we are very happy to have our guest speaker to deliver the endowment lecture on microbiome innovations for the future of food and agriculture. So my request to all the participants, so please try to utilize the facility and try to interact with our guest speaker. So before offering his endowment lecture, so let us honor our guest speaker with a shawl and
respected Vice Chancellor, Deans, Professors, Faculty, students. It's an honor to be invited to give an endowment lecture at ACNRI Madurai. And I began my professional studies and career at TNI Coimbatore. I had very rich training, learning, exposure, experiences, and also good friendships when I was a student at TNI Coimbatore. And also I spent a couple of months, maybe a trimester here as a master's student in plant breeding and genetics. Then I pushed myself out of these institutions, ACNRI Madurai. Then went to GP Pant University for my master's in microbiology. And that was my way of chasing the unknown organisms, are the ones which I cannot see with my eyes without any aid. And I began my interest, that was the beginning of my interest in microorganisms. And slowly I moved on, and I've done researches on several aspects related to microbiology, also those which are of importance in agriculture. And today's talk will be on microbiome innovations for food and agriculture. I'm going to bring in food here because we don't have to stop thinking about agriculture. We cannot afford to think about agriculture without considering what we are going to eat as food. Because agriculture, the major purpose of doing agriculture is to have a quality food. So I like to start with this question: what is life? Maybe it is meaningless to talk to all, it cannot be a meaningless thing to talk to all the biology students, because this is an interesting question. Because in order to live, or in order to do whatever we want to do, or whatever we say, like life, you know, we cannot easily define it, because it's something like blind men trying to describe an elephant. Each one of us will describe it in different ways, but life is a challenging concept to understand and then accept it. So I will just go to a book, very important book, which was written in 1944 by Erwin Schrödinger. He asked, "What is life?" and that was published in Cambridge, the University Press in 1944. And he's a physicist. He was a physicist, okay, a quantum physicist, and he was also got a Nobel Prize in physics. He asked a question, "What is life?" And he was trying to put, physics and chemistry principles related to physics and chemistry, and for the events in space and time that occur in the cell or within a living organism. And that also he proposed a concept called aperiodic crystal that contains a genetic material. And in fact, that was the idea which inspired Watson and Francis Crick to propose a model on DNA. So DNA and that time became very popular. By then, we all could understand the importance of chemistry in our life. So we could identify the elements, different elements which are considered to be essential for humans, and those which are suggested to be also essential, and those which are non-essential for humans. So we are able to, see the importance of carbohydrates, proteins, fatty acids, and other compounds of interest to us for life process. So DNA is a molecule which changed the way we look at life process. Okay, so we have now two types of genetic material, which is DNA and RNA. And this is also not only it's of important life processes, or rather information related to life process, but they also have information related to the evolution, how life evolved in the earth. So I'm not going to give you many ideas about the evolution, but again, I'm going to bring it to another book, which was written in 2021 by Paul Nurse, Sir Paul Nurse. He said his book was titled "Five Great Ideas in Biology and What is Life." Again, what is life? After 77 years of the book of Erwin Schrödinger, Paul Nurse again wrote another book, and again he asked the same basic question: what is life? And what he tries to say is these five important ideas of related to those are life process are cell, gene, and evolution by natural selection. So every time nature will make a way to evolve different organisms. Evolution is an important process. And also life as a chemistry, because most of the process of metabolic process, biochemical reactions are all chemistry. So chemistry principles, whatever which we learn in chemistry, are being applied in all the living organisms. And also importantly, life as information. So if information molecule like DNA, RNA are not available, then the chemical process are not going to happen. So gene and the information is all very much important. That's what he said. And he said, like, this is, we should also appreciate the life's richness and also their important role in biology, a role of their in our time. He says to survive the challenges that face all of us today: climate change, pandemic, loss of biodiversity, food security. For everything, we need to understand what life is. So unless we appreciate not only our life, but all of the life forms, we won't be able to understand or rather derive or develop solutions for all those problems related to climate change and then loss of biodiversity and most importantly, food security. So what was the work for which he was awarded Nobel Prize? That is also very interesting. Paul Ness got a Nobel Prize in 2001. He wrote the book, What is Life? Five Great Ideas in Biology in 2021. It took 20 years for him to write a book on what is life. And what was his work? He again, like he worked on a yeast called Schizosaccharomyces pombe, where he studied about the cell division. He identified a gene called CDC2, which is responsible for the cell cycle from G1 phase to S phase, and also from transition from G2 phase to mitosis. That CDC2 gene is a cyclin kinase phosphorylation dependent protein. And what is more interesting is that same gene is also having a similar homologue in human beings. So right from yeast to human cells, the same gene is playing a role on cell division. So for which he got the Nobel Prize in 2001. So again, to score, like give you the importance of what is the role of a gene, how gene has been conserved for millions of years from yeast to human beings. That CDC2 is very important gene as far as cell cycles are concerned, for which he got the Nobel Prize. And okay, now when it comes to microorganism, again, now we understand why microbes are very important. Microbes, they have all the genes. From there, we have evolutionary process leading to all other organisms. So these organisms, microorganisms, although we call them as microorganisms, they are not only microorganisms. There are several organisms which are nanoorganisms. Their sizes are smaller than microorganisms. And if you look at them, like some of the very common example, E. coli, which has got about 4,300 odd protein molecules are there in the microorganism. But when it comes to Pleiobacter rubicae, it has got only about 1,340 or 54 protein molecules are there. Then there is a nanoarchaeum equitans, which has got only 536 protein molecules. So the amount of genetic information which is required by this organism is so small. So you can understand, like we have now not only microorganisms, we also have nanoorganisms. So can we compare all these organisms? Like that's another important question which we have to ask. Like now, if you want to compare them by size and shape, of course you cannot make any such comparison. But but if you look at the life process, then you can make certain comparison easily. That's what Jacques Monod, who gave the concept of operand theory way back in 1960s and 50s, he also said, what is true for E. coli is also true for the elephant. So doesn't matter about the size and shape. So the process is all, it's all common for microorganism to the elephant, all the macroorganisms. So that's also like, again, you can look at the way like Dr. Sir Paul Ness, whose noble work leading to the identification of CDC2 gene. It is common across yeast and human beings. So we have several organisms, not only they live in the nature, but they are also on the human body. If you look at the number of cells in the human body, we have about 10 to the power 13 cells are there. And also we have around 10 to the power 14 microbial cells on the body, inside the body. They are all living within our body. So that's what like we have now. Like if you look at the bacteria to human cells, we have the ratio is, it differs between a man and a woman. So it can vary from 1.3 for a man, and whereas a normal woman will have 2.2 in the ratio of cells, bacterial cells to human cells. But this, and also it changes from infant to adult to the matured human beings. Okay, so there are differences in the number of organisms which all play important role in human growth and development. Why do we need all those microorganisms? These organisms outnumber the human cells, and what do they do with our lives? So they all provide genetic information. They give a lot of genes which are required by the human beings or we to digest the food, communicate with our brain, and then also decide whether we can live happily, healthily. Right? So if you make any kind of disturbances in their homeostasis or the balanced presence of bacteria and the human cells, then we'll have diseases coming up. So that is what we could see here. So we have 3.3 million number of non-redundant genes in the gut region. So because of their presence in the gut, right? So they have so many million number of genes from the bacteria, which are all in the human gut, and they actually rather they are different from two individuals even by 80 to 90%. So we differ by microbiome. What we call microbiome is all those organisms which are now living on our body and inside the body. That we call them as microbiome, and they decide like almost 90% of the diseases which we have during our growth and development. So gut is a very, very important organ of the human being. Again, gut microbiome is also very, very important because we almost carry 1 to 2 kilos of bacteria in our gut. So when you say you are 65 kilos weight mass, and you can minus 1 to 2 kilos of bacteria from you, and because they don't belong to you. But unless you carry them, you cannot do any work. Gut bacteria is very, very important. They provide 3 million genes. So they also, like as I show in the right corner of the slide, where they change because of the way you take diet, and then medicines and geography, lifestyle changes, and also like in feeding method, and then stress. All those factors, they determine the kind of microorganism you can live with, right? And if you look at the genetic basis of these genes, what is interestingly come to know is, if you look at the ancestry of human uh, genomic signature, nearly 37 genes are actually of bacterial origin. So again, it is not only microorganism. The human genome is actually derived from bacteria. So do we have similar superorganism in the case of in plants? Yes, they also have several microorganisms living on the surface and inside the tissues, xylem, phloem, root region, everywhere you can see microorganisms. So that is what we now understand, and that we call them as plant microbiome. So how do we define microbiome? This is important definition which has been proposed in 2020 by Gabriela Berg, who said it is microbiota plus theater of activity because of all the metabolites produced by the microorganisms. They play an important role. So all the metabolites, they all of them together, we call them as microbiome. So when you look at the genetic potential of plants as such, so if you look at them, like we have microorganisms present on the philosphere, on the leaf surfaces, endosphere, inside the tissues, rhizosphere, in the root region, and the soil microbiome. Okay, what do? They provide several genes to the plants to grow and develop as a mature plant. So if you look at a tomato plant, it has got only about 42,000 genes as such in the plant having around 42,000 genes. But if you look at all the bacteria which are present in the tomato plants, it will, if you consider all of them, then we have nearly 3 to 10,000 K, I mean 10,000 genes into 10 to the power 3 to 10 to the power 4 number of genes can be actually are provided by the microorganisms. So that is the level of genome contribution by the bacteria or the microorganism to the plant system. So we now call all this as second genome of plants. So that is which is interesting to us, to me rather. So if you go by this data, the paper which was published in 2020 by Wang and Hane, you can understand that like nearly 1,000 to 10,000 bacterial OTs are present, and each one of these organisms will have 3,000 to 800 genes. So we have important contributions from the bacteria. So how do we test the presence of the microorganisms in the plant tissues? So we use different dyes like cytonine, and that can give us an idea about the presence of bacteria in different tissues. Where here is an example which was published in 2008, where they show the presence of microorganism in the seed, leaf embryo, and root embryo, endosperm, and seed coat. Well, and then we also have now new methods of their contribution. not only the presence of genes, their contribution in terms of nutrients provided to the plants. And they use different techniques normally used by us for estimating phosphorus, nitrogen by chemical methods are inadequate to know about the contribution of bacterium to a plant system. So they use techniques like micro X-ray fluorescence and Raman spectroscopy combined with the stable isotope probing. All of them are used to to understand about the contribution made by microorganism in terms of nitrogen and also in terms of phosphate solubilization by the bacterium, right? And we also have a nano SIMS, that is nano secondary ion mass spectrometry method to know about the contribution in terms of nitrogen being fixed by this bacteria. So plant microbiome is having a lot of benefits derived from the microorganisms, not just by the nutrients alone. Recently, there is an idea proposed by Shauna Smith from University of Queensland, where she put up a question: do plants consume bacteria to obtain nutrients? So this is an interesting proposal where she said plants grow microorganisms in the rhizosphere as we grow plants in agriculture. Understand? So in order to consume plant products, what we do agricultural practices. Same way, plants also do agriculture, or rather, they consume bacteria in the rhizosphere region. That was an idea which was mooted in 2010, and this was again worked on by from Rutgers University, where he again showed, he proposed a concept called rhizophagy cycle. So in that, there are areas like plant cell entry zone in the root tip where microorganism will get into the plant system, and then plants will extract the nutrients from the bacterium, and then they will release the microbes out of the root system. And the microbes, when they come to the rhizosphere again, they multiply, they get into the plants. So this is what he calls as the rhizophagy cycle. So this is an important finding now, which shows about the role of microorganism to the plant nutrition. So microbiome is very important, not only for providing genes, but also for providing several nutrients to the plant system. All this made to consider an important issue about the higher organism. I would like to take the help of John Donne, who wrote a poem, No Man is an Island, that was written in 1624, where he showed that human beings do badly when isolated from others and need to be part of a community in order to thrive. So no man is an island. I take that example and to say that no organism is an island without microorganisms. So microbiome is a very important component of all living organisms. So, but they also evolve by a two-way interaction between the land or the soil and the different life forms. So, that is why you can see different type of organism coming up in different areas. So, if you look at the desert, and then there are areas which are very fertile, and those which are closer to the sea, you have different kind of microorganism coming up, and it's all because of the different evolution process. And this nature of evolutionary process and the life process, we are changing it, because in the last 12,000 years, we have been, deforestation, and for the purpose of growing crops, we actually clear many lands, and then agriculture is almost now is an important, or rather very significant factor for changing the way the nature in the whole planet. So that is what we call them as now andromes. So we are now developing new environmental ecological system, which we call them, which are influenced by human beings, we call them as andromes. And if you look at the way the andromes are developing, and these are the ecological patterns shaped by human activities, largely by agriculture, and that is why we have a different human biomes or androgenic biomes. If you look at India, you can see that most of the land mass is now completely transformed, and we have a lot of andromes interactions. So if you compare between 19,000 and then where you can see India is having very few regions in pink color. And then now if you look at them, all of them are now changed to rice, irrigated land, rain-fed land, and then pasture land. So those so much of intensification of agriculture and land is changing. And the way urban areas is growing is also changing. And if you see in the last 300 years, you have many cities are coming up, urban areas are coming up. So the challenges for agriculture is too many, and land transformation is the biggest story of biosphere so far. And I'll just show you two examples, although many of you must be knowing about the climate change events, global warming, and sea level rise, and all those things. Recently, there's a paper in Geophysical Research Letters where they showed that because we pump out the groundwater, mostly for agriculture and human consumption, which is around the 2,150 gigatons. Now we have a change in the way Earth is rotating. There is a pole is has moved. Rotational pole is completely changed. So you can imagine the amount of the change we are creating by clearing land areas and then transforming many of them and doing agriculture with a lot of groundwater withdrawal. Right? That's an important one. Another one is antibiotic or antimicrobial resistance, antimicrobial resistance. So when we start using different antibiotics as a INFS for increasing the poultry farming, and also like we have several what you call use of antibiotics across the system. So we have now resistance developed by simple bacteria, and those are now contributing to the development of antimicrobial resistance. This is now spreading very alarmingly across the earth in different ecosystems. These are the two things which you also should take note of, because these are all going the way we do agriculture. Since most of us are from agriculture university, I'm giving some of these examples so that you can understand like how do we make changes and how to think in a new ways. This is another example. If you try to do agriculture the way we do now, business as usual, so we have so much of pressures on the natural resources, not only water, also in the plant and in the land and the plant diversity, so genetic diversity. So that I will show. So the current model of more production is outdated and inadequate to our needs, because we are even then, even after intensification, so we are unable to meet all the needs of several people, and ours is a very highly populated country in the earth now, and we have more problems to the environment and natural resources because of the current model of more production to meet the demand of human beings. So we are, if you have that model, produce more to feed the people, then I'll say we are aiming at the wrong goal. We should now understand like what are the new better production systems, so that, and also better food systems. These two are important so that our agriculture is meaningful and then it is environmentally friendly. So we need to understand about like what we need to do in the future, that we should have some foresight and also to plan for the future, because the model of developing several, I mean, current model of producing more is going to give more problems to the environment. So foresight is a process of visioning alternative future through hindsight and insight and forecasting. So I'll say we need to define the goal of human nutrition in terms of human nutrition rather than on more production. The reason is, so this is what we all have been taught for a long time. So in order to keep ourselves healthy, we need to eat the right kind of food. So what we eat, like these, whatever we eat, should supply us carbohydrates, proteins, and fats, and also minerals, and then vitamins. So this is an example by which we have been taught to do, or rather we have been motivated to do all our agricultural activities. But if you look at the proposal or the idea presented by National Institute of Nutrition, you can look at it like we need to have about 350 grams of vegetables and about 150 grams of fruits on your plate every day. Anytime you have a meal, you should have 350 grams of vegetable, 150 grams of fruits, and then about 240 grams of cereals, different kinds, and pulses. So next time when you have your plate, you just see whether what is recommended by the National Institute of Nutrition and what you see in your plate are comparable anyway, right? That's well, what is missing here? Well, I will say there are several things which are missing. What is, I mean, to highlight few of them, we have been considering only 6,000 agriculture and horticulture crops, although plant species are running around, you know, I mean, I'll say 4,000, no, 400,000 vascular plants are available, reported. And about 20,000 or 28,000 are edible plants. But among that, like we only know about what we have been taught or what we are now teaching about production methodology or agronomic practices for about 168 crops. Again, we focus more on 9 of them only. So our focus is completely out of the diversity. So we are focusing more on just about 9 crops to have more carbohydrates, proteins, and all other needs, vitamins and minerals. So we are missing out the diversity, and we have been focusing more on variety of those 9 crops. Is it the right way to do? You decide for your future, because most of us have almost lived our life, and it is for you as a student to think about like what you want to do, at least in the future, right? So again, that is, I mean, although I've been telling you about the microorganism in the gut region, where do you think like these microbes are coming from in the gut? You think they have been developing in the gut on its own? No way. We have been giving them in terms of different food items. If you start cooking all of them, cook them or fry them, and then do whatever you want to do. And if you don't give any microorganism, will it be healthy? Doesn't matter. So our food is a major source of microorganism in the gut, and which we have forgotten. So all our food which are without microorganism, live microorganism, are going to be, will be the major source of several other complications. Many of you may be knowing about the non-communicable diseases like obesity, diabetes, and cancer. And all those things are largely because we forgot to include microorganisms which are necessary in our gut region. So if you look at some of the recent papers, they are an interesting proposal to say we and herbivores eat endophytes. Those who are having microorganism in that food are are doing better. Okay, and there are now connections to show that like the healthy soils are important for healthy plants, and also healthy plants are important for healthy humans. So your soil is an important one, because microorganisms which come from fruits and salads and vegetables, they join the human microbiome in the gut region, and they will help you, I mean, keep you healthy. That's very, very important for our life. Okay, so in some of our experiments which we do in my laboratory, we focus on the rhizosphere region to understand like how different soil types contribute to the plant development, and what are those, and whether are there differences among different soils, and what their contribution in terms of microbial species, and also the genes related to several functions related in carbon cycling, nitrogen cycling, and other nutrient cycle, and also some unique, traits related to plant growth and development. Here I'm showing you some examples where when we compared 3 different soils using metatranscriptomic analysis, we could see that like the contribution of microorganisms from these 3 soils are different. They are not same. So although I try to grow these 3 type of soils, the microbial association with those plants are different in 3 different soil. That is the reason probably we get the kind of changes we see in the food grown at different regions. So this also we should understand. So I'm showing you another picture where again to show you the importance of the microbial contribution from different soil types using metatranscriptome analysis. I'm not getting into this because nowadays many of you will be slowly will be understanding about the role of metagenomic analysis, metatranscriptomic analysis, metaproteomic analysis. metabolomic analysis to understand like how microorganisms are contributing to the life and growth and development of plants and human beings, right? So if you look at the human gut and plant roots, again, there are several commonalities. The microorganisms which are present in the gut region and their traits, and also those which are in the rhizosphere, they show a lot of commonalities, which are very, very interesting. All those things we are able to understand because of the new methods like metagenomic analysis and metatranscriptome analysis. So root, the plant root which you pick up next time when you pull the plant from the soil, if you look at the roots, you can say that that has got something, a lot of similarities with the gut of us, gut region of us. So we have so many functions of microorganisms which are common in these two regions. Well, we also have a broader understanding or a bigger perspective on the microbial diversity. There are several papers, of which I will say the paper by William Whitman, which published in PNAS, which is very, very interesting because here he showed the mass or the reservoir of or amount of carbon stored in microbial cells is runs into 350 to 550 petagrams, 10 to the power 15 grams. That is almost about 60 to 100% of plant carbon. So whatever carbon which is present in the plant on the earth, the same amount of carbon is almost present on the soil by microorganisms. That is the level of diversity we see. And if you look at the way they have other nutrients like nitrogen, 85 to 130 petagrams, 90 to 14 petagrams. So that amount of nitrogen and phosphorus is there in the microorganism. These are the elements or the nutrients which are cycled by the plant by the microorganism and made available to the plants, and they are the one reason important. Okay, and the microorganisms have a turnover time of 60 to 25 days, and they are turning over. So because of that, we are able to have many nutrients made available to the plant for growth and development. And also we now know that there could be 10 to the power 12 or 1 trillion microbial species on the earth. So that doesn't mean that like we are not disturbing this organism. The number is too many. So we have an assumption that these microorganisms are too many, so they will be present in all the ecosystem. Not necessarily. And this is one example which I wanted to show. After the process of fixing atmospheric nitrogen and developing Haber and Bosch process for preparing nitrogenous fertilizers, we started applying urea and other nitrogenous fertilizers for increasing the productivity of many of the crop plants. And that also is important because it has made a very fundamental difference, in deciding about the feast and famine of several regions. So we made food cheaper, in the sense we made it available to human population, and then that was after the discovery of Haber and Bosch process. Because of that process, we are now able to feed several people. Okay, so that made all the difference. And if you continue to do that, you know, and what happens to the microbial genes, or I mean microorganism in soil? In our lab we looked at the distribution of genes related to nitrogen fixation, ammonia oxidation, then denitrification, and archaeal ammonia oxidation. We could see that nitrogen application is having a stronger role on the abundance of these genes, microbial genes related to nitrogen fixation, ammonia oxidation, and denitrifications. So when you keep adding more amount of nitrogen, the nitrogen fixation is actually getting lesser and lesser. Ammonia oxidation is getting more and more. So all those things are now being realized. Like now we start applying nitrogen in a heavier doses. And if you look at the way the nitrogen deposition occurs over the period, look at the top left in 1816. The yellow color in India is very, very less application of lesser rates of nitrogen. And then in 1990, you can see the pink and then light pink color, and where you have more nitrogen applied. 2014, it has crossed 2,500 to 5,000 kg nitrogen per kilometer per year. So it is way beyond the level the soil can take up. Since if you keep more nitrogen, keep giving more nitrogen, you are going to lose several microorganisms from the soil. This one example is going to, you will be able to see it, at least in your lifetime. And I'm able to see it now in 2014. When I was a student, I never knew that nitrogen fixation is going to be, slowly disappearing from the soil, which I never expected it. Well, so we now depend largely on industrial agriculture to feed the population. So we have been using a lot of fertilizers, pesticides, and all of them are having stronger effects on the diversity of microorganisms. So again, you can compare the situation of land management in a normal system and industrial system. In an intensive system, we are going to lose several microorganisms, not only microorganisms, all other organisms related to the diversity. Because in sustainable system, we have a high diversity. So that will all have an effect on the human health. Well, you can, this picture, the content will again show you how this is going to directly affect the diversity of microorganism in the human health. Again, this is for you to appreciate. And not only just diversity, microorganisms are also involved in several other processes related to the, decomposition and then respiration, remineralization, and the emission of different greenhouse gases like carbon dioxide and methane. So all of them are now changing. So microorganisms play a larger role in climate change events. So although it may appear like climate and then there are no connections, but actually every small microorganisms in the soil and environment, in the water environments, are all getting affected by the application of fertilizers, pesticide management, and intensive farming systems. All of them are making changes, and they also make larger impact on the distribution and composition of pathogens. And this is again, climate change will have a serious effect. So if you don't see that, this is what we will go and end up. One day we will suddenly see that everything is beyond our control. We cannot make any changes. So agriculture, as agriculture graduates, students and agriculture scientists, it is our responsibility to see what will happen if we continue to do what we do now. Business as usual may not be that much efficient way of utilizing the natural resources. I'm giving you another example, which may be easy for you to appreciate. So apple, when you eat apple, we think that apple in a day will keep the doctor away. So we think that way because the contribution of apple in terms of nutrients are often considered. But interestingly, apple, when you take them out, like if you have a 400 gram of an apple, then that will have about 100 million bacteria. So when you take a raw apple, you are not only taking nutrients, but also having 100 million bacteria which is going inside. So that's an interesting thing. And you see that, like, the contribution of different organisms in stem, seeds, fruit pulp, peel, and then all those things be now worked out. So we understand how these bacteria are present in different components of the apple. And apple, which is grown in Israel and US, and that is grown in Turkey, they're also different. The Turkey is having more fungal organism, whereas those apples which are grown in Israel and US are having more bacteria. So all those differences are now understood. And if you grow apple in a intensive farming method with the use of several fertilizers, application of fertilizers and pesticide, you're going to lose several of these bacteria. 100 million bacteria is not 100 million bacteria. You are going to lose many of them, not only in terms of numbers, but also in terms of the composition. So they may have a different set of organism in the industrial farming method. Those apples which are from the orchards using these fertilizers and pesticide will have a different composition, whereas those which are in the organic farming will have a completely different decomposition. So now we understood, like we are now trying to understand, or rather, we know the contribution and benefits of microbiome in terms of health attributes of the fruits, giving out the carotenoids, and then bacterial volatile ion, key metabolites, and all those things. And how do we those factors related to how several other determinants play a role on the composition of microbiome of an apples. Okay, so we also now we have a fairly good idea about the post-harvest losses. Not only the method of producing apple or any other agriculture product is playing an important, important, but the losses which occur during the processing is also playing a role in the availability of food and distributions. Okay, and microbiome can come into our help if in order to understand about the losses, because now we know we have methods like what I described to you earlier. Metagenomics, metatranscriptomics, and metabolomics methods can be used to know like how the losses are occurring, right? So this is our now microbiomes are microbiome is used for tracking in the sense we understand like from the one orchard to the final consumer, from farm to the fork, from farm to the table. So you can track them, like what kind of losses which occur and how apples are getting, quality changes occur because, and what are the factors which contribute to the those quality changes by tracking the microbiome composition right from the farm orchard to the consumer's table. So that is possible. So now this is being used for determining the quality of products, different products. Also, I've been talking about like how the current methods or intensive agriculture uh, are actually having effect on climate change and also on the quality of life. We now need to look at like how to go for a method by which we can have a diversified agroecological farming, which is not to be like industrial agriculture, not like subsistence agriculture, but again, considering the limitation and the benefits or strength of the ecological system, one can develop new food systems. So again, there are a few methods which are coming in now being recommended. One is regenerative agriculture. Regenerative agriculture is basically it's a an idea which is proposed so that we can rebuild all the soil health related parameters and also soil microbiome so that the soil is able will be able to support the plant growth. Soil is fertile. So regenerative agriculture again, it's again going to depend on the on microbiome traits and microbiome innovations, which will actually will make the soil fertile for a longer period rather than their dependence on fertilizers. And there are many ways we can, employ different microbiome or microbiota in the production system. Here I'm saying something like microorganisms are now applied not directly to the soil. Maybe you can apply them on the foliar regions and seeds. Also at the time of flowering. So there are new ideas which are being now proposed for delivering the usual or delivering the beneficial organism to the plants. All of us must be knowing about the seed coating of beneficial microorganisms, which I've been doing in the division of microbiology for several microorganisms like Bacillobacter and phosphobacteria and other things. So there again, we could show that like if you apply these useful organisms, beneficial organisms, they are able to colonize the plant system and then they benefit the plants. Again, now there is a new method where you can directly apply microorganism at the time of flowering, and that will get into the seeds. So instead of coating the seeds, you can deliver the microorganism at the time of flowering. So that is being proposed in 2017 by Mitter and the laboratory of Angela Justice and the group, and they now come out with a patented method called EndoSeed, where you can deliver microorganism at the time of flowering, and they are able to deliver microbes to soybean, maize plant, and then pepper plants. Here, when you use fish and confocal laser scanning microscopy, you are able to see the organism which have been delivered at the time of flowering could also be seen in the seeds, right? There's also another method where microorganism can be directly injected into seeds that they call as seed injection. Okay, seed injection. So this is being now where you can directly insert into the endosperm of the seeds. Seeds have to be a little bigger because you cannot do for all kind of seeds. But of course, this is being now commercial, like it's in the different. These are the different methods by which we can deliver the beneficial organisms. Again, I also want you to consider microbial foods for improving human and planetary health. We have left out several foods which are directly based on the microorganisms. And we grow several organisms, and fermented foods are very important component of food system in the past. So we are slowly losing them out because of our preference to the processed food item. Most of the processed food which are available in the commercial e-commerce or wherever we do in the marketing nowadays do not have sufficient microorganisms. Fermented food like although idlis, although it's a fermented food, but we steam it, then we use it. Or any other fermented food like rice which is taken after overnight fermentation, and that is an important source of microorganism in the past, and now we have left them out. And recently, like in recent times, we have kombucha. It's also a fermented food, which is now becoming popular in US and Europe, and that it's almost now touching the market value of 1 billion. So anywhere you go in the US in supermarkets, you can always buy kombucha, which is nothing but fermented food. So we have left out. I mean, we haven't gained from these kind of fermented foods in India, right? So now again, as with all other professions, we also have to consider about the ethics. So what are ethics? Ethics, when you look at it, there are four principles which are important: do good, don't harm, respect, and act justly. In the current model of more production in agriculture, do we do all these things? Ask yourself, not by the local level, but at the global level. Are we doing justice to the earth? Are we doing good to the earth? Do we do harm? All those questions. Because we have never been taught when I was a student here. Okay, we are all taught how to produce more, how to produce more. And that, like now, it is time for us to think again. Can we use the same model to sustain human population for the future generation, right? So again, there is an interesting paper which has been published in 2022 in Environmental Microbiome, where Burke and Brooke Lang they said something about microbiome ethics. There again, they say in order to have about the diversity, don't do harm, do good, respect and protect nature's biodiversity as heritage for the future. So we focus on 9 crops for all our consumption. What do we do with the rest of the plants? We clear them off, remove them totally, then we grow only 9 food crops. What we do with all other animals? We have chicken, meat, and rest of the animals are gone. How many of you can count number of tigers in India? 200, 300, 400? How many cheetahs in India? How many lions in India? Less than 1,000? So we are losing several of them. So diversity, yes, protect nature's heritage and diversity as a heritage for the future. And soil, do good, don't harm, and do good by improving microbiome for soil health and soil fertility and soil remediation. These are some of the principles which we should also think about. We should have in our mind when we plan for our future, when we work for our future, right? So there are some principles which have been listed by the same authors. Again, I'll say microbiome research is one of those priorities. And they say this is going to help you in forecasting, in planning for the future, right? So this I will leave it there. Again, food fraud. Many of us must be knowing about the use of chemical adulterants in our food. What do? Because it's a making attitude. Some people wanted to take advantage of others' ignorance, not knowing how, and they are introducing several chemicals. can we use microbiome tracking for understanding about the food fraud? Yes, we can do. Microbiome forensics is being used to understand about understand about human loss. Forensics principles are being applied. Same tracking forensics on similar only. So microbiome tracking can help us to detect food fraud and also to develop regulatory methods for understanding about food fraud. Again, we are now getting into another era where we think about food in terms of blockchain and then artificial intelligence and then also using different packaging material so that e-commerce will benefit largely from the agriculture products. Okay, so again, there is one such idea which is now becoming popular is 3D printing of food. All we have to do is we need to have a paste of different components, semi-solid paste of several components, and printing can take, you can produce chocolates, cookies, and several other things. And also you make what you call fruits and vegetables by 3D printing. So these are all becoming possible. So in the future, if you wanted to check the quality of 3D printing of food, well, microbiome can help you. And this is one example where they are making chocolate using 3D printing. All you need is a syrup of chocolate and different components, and you can decide what you wanted to make. Same way, like if you decide about different components of fruits, banana for the matter, you can make a 3D printing of banana because you don't have to preserve the storage of bananas. It's going to be really difficult because you cannot store for a longer time. But if you make a powder, and then the powder can be reconstituted like a baby food, and then the banana can be created by 3D printing, food printing. It will become easier. No, you can always get a feel of banana, and then you can also make use of banana for a longer period. I mean, to sum up, like I will draw a few examples to say there is no cheap food. It's an illusion. So to say is the words of Michael Pollan, there is no such thing as cheap food. Whatever we make, whatever which is being made available to us, if you consider them cheap, but the real cost is paid somewhere. It is not paid at the cash register. It is charged to the environment, to the public purse in the form of subsidies. It is also charged to your health. So all the methods, current methods, are when you look for a cheap food, it's going to affect your health and at the global level. And I have another word to share with you because although we produce more, and also we waste more and more. So to give an example, they say if whatever food which you are wasting the last every year almost we waste around 20 to 30 to 40% of the food which actually we producing food post production losses processed food. So if you take 40-ton trucks and then the amount of food which is wasted it will be around 23 million. Okay, and if you allow the trucks from bumper to bumper, then it can make a 7 cycle of an earth. So that much of food is getting wasted annually. So that is a level of waste which we do. So all those things can also be changed to useful products if you make use of different fermentation methods and different products by using microorganisms. Also looking like you should have new innovations to make use of them rather than wasting them. Again, I will also share another concept which is being now proposed is what do we like to do? I mean, if you wanted to keep your earth good as well as sustainable for a longer period, this is your plate. You should have more of. I could not hear you. I'll talk to you later. Okay. They have more fruits and vegetables, and not. And if you look at this planetary food plate and the one which is being proposed by National Institute of Nutrition, I've got a lot of similarities. Can you see them? So not like all your cereals, rice-based food or any other cereal-based food, but it's mostly about the fruits and vegetables. Well, these concerns are also, you should not only global warming and what Stefan and Rockstar, these are the two authors who have been proposing about the boundaries the earth can sustain the life. and when they define the process which are actually cross the boundary limits. And some of them like biogeochemical flows related to nitrogen and phosphorus application. These by application of chemical fertilizers, already we have gone into the red zone. So planet cannot take more and more fertilizers, cannot in terms of nitrogen and phosphorus. And planets are also losing large biodiversity in terms of integrity is lost. So several organisms are now slowly getting what we call endangered species. There are often there is a list. UN is coming out with a list of endangered species of animals and plants. Soon we'll have a new list coming up for the microorganism also. And there are now, earlier we were thinking that like there are so many trillions of microorganisms in the earth, and we don't have methods to detect their loss. But now we know we are losing several microorganisms. So these are also of a great concern to us. And well, to end again, I'll take you to the same old story of blind man and an elephant. So what is life? So our perspective, our understanding of life is limited to our, the way the six blind men describing life. So if we are all so focused about, each one of the different parts of the earth, we may not see what is happening in the whole earth. So that's what they are saying. We have limited experience, which means we have a partial perspective. So we don't remember this limitation. We can get into trouble with ourselves for others. Elephant represent many different things in life that we cannot see, and our senses are limited information. So it is better for us to understand these limitations and also plan and behave. Well, I thank all my students, my teachers, and all my colleagues in my journey of understanding about the way we do agriculture to sustain life forms on the earth. Well, again, I will say I thank you for patient listening.
This is Sivakumar, sir.
Yes, Dr. Sivakumar.
How are you, sir? I'm sorry
Fine.
from the beginning I was unable to attend, but second half of the lecture was very excellent. There was some connectivity issues here, and some of the
Sorry.
topics like soil to plant and plant to human health. So all these connectivities are very excellent. And one thing that was interesting was seed section.
Yes. Yeah.
Uh, can you give me, please?
It's a new technology which is being proposed or being patented by Dr. Mitter of AIT, Austria Institute of Technology. And Dr. Mitter's paper is available in 2017, Frontiers in Microbiology, and her email is there. And
Okay.
she is now proposing, or rather, she has patented this technology. It's good for seeds which are having a bigger endosperm because after injecting bacteria of interest, they have to be sealed again, and the cost of seed injection will be very high.
Mm. So seed
So
injection means seed coating or seed imbibition.
it's
I'm
not.
not
It is
sure
not.
course
It
which
is
actively
not.
injected or passively.
It is not seed coating. It is different from seed coating. It's directly injecting into endosperm. It is like gene gun method, which is being used by geneticists for delivering gene into plant tissues or cells. In similar manner, microorganisms are directly delivered into endosperm region tissues. Okay.
Okay, sir. So that means this will travel throughout the plant.
No, after it is injected into the endosperm, when the seeds germinate, they will travel to the.
So this is Kalachilvi.
Yes, Dr. Kalachandi.
Sir, it was a very good eye-opening thing. I have a small
Hmm.
doubt, sir. Suppose we have more than 5, 6 organisms
Hmm.
of endophytic nature. So how to go for seed treatments, like seed coating or seed treatment and other things?
More than 5 to 6 organisms. Delivering all the organism at the time of flowering will be the easiest method in the future.
Okay, sir. Okay, sir.
because although we cannot say whether all the five will equally colonize the seeds, but
Okay, sir.
delivering them all at one time during flowering will be the easiest method. Seed coating will be difficult again because seed coating again will create a competition with the rhizosphere bacteria, and they
Okay,
may
sir.
not be able to get into the plant system. But whereas flowering will directly take them into the plant system if they are compatible with the plant.
Okay, sir. Sir, is there any like dose or inoculum
Well,
load?
we have been okay. Now I have a student who is working on delivering beneficial organism directly into the rice plants at the
Okay, sir.
time of flowering, and we are only tracking the presence of these delivered bacterium in the subsequent generation. But we
Okay,
have
sir.
not worked on the number of or the population density I can deliver into the flowers, because rice is a self-pollinating crop and it is very difficult for us to deliver. But in the case of cross-pollination, yes,
Okay,
it
sir.
may be possible. And well, that's what honeybees were doing in the nature in the past. We killed most of them are killed now by pesticides. So we have to now physically deliver beneficial organism to the plants.
Okay, sir. Thank you, sir. Thank you. Thank you so
Thank
much,
Sir,
sir.
you.
good afternoon, sir. Good afternoon, sir. This is Jnana Chitran, sir. Uh,
Please.
sir, yeah. Sir is telling during flowering is the best stage for application of bioinoculants. No, sir. But
It is
we
one
are
of
applying
the
biofertilizers for mainly for nutrient acquisition.
yeah.
And this nutrient acquisition happens, that depends on the basal fertilizers which we are applying at basally. Okay, sir. So the bioinoculants are very important at the beginning stage of any crop, no, sir.
I have few issues to be discussed here, because if you are delivering organism at the time of flowering, and if you try to add fertilizers at the time of germination or basal application, and the organism may not do good to the plants, because microorganisms will become sensitive to the fertilizer which are being applied, and they may not do. But if you allow them, like there may be a low concentration or low doses of fertilizer, then applied organism can easily colonize the seedlings during germination, and they can do good. But later stages, if you want the nutrient acquisition to be better, early colonization will be better. Okay, so these are not being worked out. I have no idea. I'm just presenting the new, innovations or new ideas which have been proposed by the group of AIT, Austria Institute of Technology, which I think we should focus on, investigate, then do it for our system so that we can minimize the failures of seed coating.
Sir, since we are working in this field, and sir,
Yeah.
bioinoculants is mainly for the increasing the fertilizer use efficiency. No, sir. That is why we are applying at the basal, that is through seed treatment or through soil application. Maybe foliar application at the time of flowering also good, then, sir. No, sir. It is my doubt only, sir. I'm not sure.
You may do the
Mainly.
experiment and tell me the results. I will be interested be interested to know.
How many generations can we take this through the seeds and flower application?
I think right now they have seen up to F2. Like 2 generations.
Generations. Balachandra is working on that.
Yeah.
Student has completed
This is okay.
thesis. Price.
Andres.
Yeah.
Okay.
She has completed one generation. So,
In my case, my student has completed up to 2 generations. We are able to see that. And well, we asked her to go for the 3rd generation. 2 generations, yes.
Sir, is it using GFB? It was tested, sir. Green fluorescent protein, it was tested.
Yes, my colleague Dr. Archana Suman, she has tested using GFP tagged organisms in
Okay,
corn
sir.
plants.
Okay, sir. Okay. Any other methodology like we can go for tagging?
Using qPCR as well as metatranscriptomic analysis.
Sir, good afternoon, sir. I am G. Tangning, sir.
Yes.
Sir, I had a question regarding seed injection. Regarding which microorganism has been employed for seed injection and which crop? And what is the output, sir, in terms of yield, or what is the expected outcome, sir?
Expected outcome is, there are all desired changes in the plant traits related to seedling growth and then leaf greenness or chlorophyll content. So this is what they have seen in corn plants. And then what they have used is they used the Paracannabis organism. These details are given in the paper of Mitter, published in 2017. Okay. And seed injection, she didn't give many results. This was shared by one of our colleagues, Dr. Stefan Kampant, in a recent meeting in Seattle. Okay. I happened to attend the meeting, so I was curious to know what kind of a gene section they are doing. Okay? So I thought like we can also test in our universities and institutions. Okay.
Yes, sir. Sir, I also had more doubt, sir. So whether there is any significant change in the population count in terms of seed injection, whether there is distinct changes in the population or a significant effect in terms of increasing the population of the endophyte.
The numbers are not being quantified, but there are changes in terms of composition. So if you look at the microbiome composition of injected seed and the non-injected seed, there are differences. But that difference cannot be directly related to quantitative information. So composition is only, it's only a comparative method. So right now we don't have information. But using certain biomarkers or several other groups of organisms, one can do quantitative analysis. Right now, these data are not in the public literature. They are all trade secrets. They are not going to share it with us.
Yes, sir. That's sir. And one more question, sir. Whether there is a whether it has been compared with the conventional method of application regarding you have told that there is difference in the composition of microorganisms. Whether it has been whether it has been compared with the conventional method.
What is conventional method? Seed coating?
Yes, sir.
So it has not been compared with the seed coating or seed treatment, but it is compared with the non-applied or non-injected seeds. Okay.
Okay, sir. Okay.
So you have mentioned about kombucha, sir. So it's an fermented which is originated from China, sir. So it has either bacteria or yeast, any one culture only in that. So, but yogurt has even more probiotics and compared to this, sir. But why haven't you mentioned about that?
Yogurt, yes, there are some yogurt is dairy product. Okay. Dairy product, right? Kombucha is a direct drink. I mean, we're using bacteria and yeast, which you use it. So dairy products, there are some issues with the dairy industry now, because dairy industry contribute more to global warming, and dairy, you know about that. So we have more carbon dioxide emissions. So people are now slowly moving from, what you call vegetarianism to vegan, where they even avoid dairy products. So in considering those facts, like I didn't want to share about the dairy product. Yes, yogurt is good. But if you ask any of those, Siddha the doctors here, they will say more than yogurt, you take buttermilk. They will not say you take curd or yogurt in the form of curd. They will say you take buttermilk, because that is considered to be the best for human health rather than taking curd or yogurt. So it is, you will have to, try out and you find out which is good for your system. Because microbiome of you and another person, me and anyone, everybody is different. If you are able to accept it, and if it is doing good for you, follow it. There is no uniform diet for everybody. Yes, thank you. I was trying to answer. I could not hear. I thought if you have a question, I will answer. No,
as they having a probiotic activity in fish, have the same role in another organism.
You can answer. The
bacteria Strobacter TNT, probiotic, bacteria and fish. Whether the bacteria plays a probiotic in a stingless fish.
we can't say that unless we have any strong evidence to say that. Because we can't take that. Although I know in several industries, what we call prawn culture, they try with the Actobacter and then Azospirillum organisms. But I will not agree with this because unless you have data, you cannot say this. So the have to become different because fish and another animal going down the different. Although we have a general understanding like lactic acid bacteria, so they are good for every organisms. So generally considered to be harmless, beneficial, but cannot be.
We are applying microbes the flowering stage. Whether it can affect to the pollinators or for the social insects.
So once it gets into the flowers, it cannot affect the pollinator. What we can learn is like you understand what are the bacteria which are being delivered by honeybees. Then you can deliver the same things to the flower. To tell you another interesting story. California is having almond trees. US. At least though I know in Maharashtra also they have an incident. So those trees are cross-pollinated, but California, that region doesn't have enough honeybees. So what they do is they bring honeybees from Florida, Florida and other regions. Then they take it to California and release the honeybees so that walnut trees flowers are pollinated during the flowering time. Same thing I could also see. There was another news in the newspaper in Maharashtra. So when they started having honeybee hives in the farm, and their farm productivity was better, higher, and farmers are now having honeybee hives. So even in our own country, there are examples. So from those organisms, if you know what are those microorganisms which are delivered at the flower, you can do that. So it is all. I mean, we should at least accept the fact, appreciate the fact. This honeybees pollination is not only for pollen growth or pollen exchange, but is also for taking the bacterium from one flowers to the another flower, then giving the microbes to the.
Sir, won't it become GM foods?
Which one?
The microbes get transferred from one plant to plant.
It is not genetically modified.
Microbes may do the horizontal transfer of genes.
Yeah, it can do. So these are all naturally occurring causes. They are not tinkered by us. We assume that we know better than the nature, and we try to clone the gene from some other sources, try to put it in a system. But in the nature, if doesn't permit, and it will spew out the DNA.
Then this endoseeding, sir.
Endoseed, yeah.
By nature.
Endosite is, again, we are collecting the bacterium from the nature only, and we are introducing them to the system. And if the plants do not accept the organism which you select, they will not have in the seeds. They know the ways of keeping those organisms out, and the composition will not allow the other organism to get inside. So they will have a competition. But considering the competition which you normally see in the rhizosphere, flower is less. The competition of introducing a beneficial organism in the rhizosphere is tough for us because the competition is very, very high in the rhizosphere region. But in the flower region, very less number of organism, composition is limited, less abundant of other organisms. So the competition for the introduced organism is lesser. So that is one logic by which we have to work. So rhizosphere, we thought like nutrients are better, they will do all those things. Now we can also think about the introducing in the flower.
So I have a doubt related to the bird tube sir. How you are selecting the microbes for floral applications, sir? As you said before, plant it will select the particular microorganism inside. Then it will be specific for each crop, sir. For
There are
writing.
two ways microorganisms are selected by the plants. One is by, what you call what they travel inside the plant system from the root region to the flowering region. So then when they are transmitted to seeds, we call them as vertical transmission. Okay, so that is a unique set of organism. But there are other organisms which are picked up by the wind and by the interaction between plants. So those we call them as horizontal transmission. So that those two modes can be used for the delivery of organisms. So plants has got certain factors. They will allow certain organism to be picked up from the horizontal means and the vertical means. If you select those vertical means, they are going to be easily accepted by the plants. Horizontal means we may have to again test several organisms and find out which are useful for horizontal transmission.
For each crop, we have to select
Yes.
based on.
Yeah. So one tablet for all the disease. We tried in the past thinking that one organism will do all the job. No, it doesn't. We should accept that. Like that is why, like we understand now, all the plants having a different groups of organism, and they use this microorganism as a source of gene in the times of their need. Whenever they want, they will collect the genes from those organisms which are useful to them. But they will have a large pool. Like you have a lot of money in your bank, but whenever you want to spend, you will take the money out. The same way, they have a bank of genes available on the whole plant system. And they will use the genes which are required at a particular stage. They pick up from those bank and then use it. That's simple, I'm putting it, but the interactions are very complex, which even I don't understand. Okay. To be honest, I can say that much, but that's the way I look at them now. So why do they have so many organisms? It is like like we have 10. We have say about say in the case of tomato, I said 42,000 genes are there. Can you make all the 42,000 genes functional or expressed at all the time? They don't do. Because if you look at 42,000 genes, and the root region will select few genes to express as root in the root region. In the shoot, some other genes will be expressed. Leaves, some other set of genes are expressed. In seeds, flowers, all unique sets are expressed. And if they want to use suddenly some genes which are required for certain function, you think they will go back to root expression? No. They will take it from these fellows, microorganism. They'll say, okay, give me some genes like that. They may collect from them. That is the reason we call them as second genome. So they make a selection. Okay, this set of organism can be in my flower region. These can be on the leaf surface. This can be on the shoot. Like that, they collect the bacteria which are useful to them. And that is what we call them as balanced or homeostasis condition. So if you disturb them, by fertilizer, pesticide, then we have a different difference. I mean, there are some problems related to their relationship, right? If it affects the physical parameter, there is no point in doing it. Okay. If the parameters are affected after injection, like seed germination, seed viability, if it is lost, we should not do that. So there are few type of seeds which is having a large endosperm where even if you make a small injury, doesn't affect the seed germination parameters, then it is okay. And these injections have such a minute needle and they will not do the harm. And there is a filler also used for it. These techniques are, to me, it is new. I'm presenting it to you so that you can develop an interest in, by the time you mature as a professional microbiologist, you will come to know, okay, these are in the market. So then you can try to apply it, or rather you can develop your own application method by which you can deliver the microorganism. So we never thought about injecting microorganism into the endosperm before. Right now, these ideas are being tested and they found to be useful. So you can always do experiments and then standardize it. Then you develop a method. Okay, it is left to you to take the idea and develop it into a product. The ideation is being presented here. So what I say is methods which I never knew before are now coming up. So it is for you to for us to test.
Good evening, sir. I'm Akash, passing first year MSc Mathematics. And you talk about 3D printing, sir. Which is very nice. I have never heard. And I want to know what type of devices and machineries are used in this. And I want to know whether it's eco-friendly or not.
Eco-friendly in the sense, that is the reason. 3D footprints are interesting to us because it is like if you go to a market nowadays, want to buy color, paint, what do? You think, in our times, earlier times, they used to have a buckets of different colored paints, and they'll say these are different colors. You have to make a choice and do. But nowadays, if you go to the, color paint shop, you want to buy color, he will give you a flyer or a pamphlet and ask you, you select a color, we will do the mixing and give it to you. So same way, if you have all the ingredients related to making a chocolate, making some other product, like as I said about banana or some fruits, for all the components which are required for making, and then you do the printing at the time you want to buy it. Is it going to be useful to you? It is going to be useful. You will soon realize it. Because if I want to have, say, an apple, and now what we do, we are actually doing a lot of waxing, coating of apples and preserve them in cold storage. And then we have a certain limitation in number of days they can be stored. But if you make a paste out of the pulp and the peel in another container, you bring it to a 3D footprint, and you design it in such a way that you give a command for making apple. And if it makes an apple, and then you can make, will it give you an advantage or not? It will give. Maybe in the future you will come to know, because that will save the storage limitations of all the apples in the cold storage. All you need is a different containers for pulp, and then for your seed coat or like fruit coat. You make it and it will give you an apple easily. So it is imaginary. But what I see in recent time is this 3D footprint is being used for making something like a burger. And all those things are now coming up. So it is a futuristic idea. It will become possible in the future. So all you have to do, you don't have to go to the fruit market or fruit stall to buy things. So get into your 3D footprint and then ask them to make apple, then it will be maybe a possibility, which you may come to see it in another 10 to 20 years. But there again, you have to see the quality of organisms which are present in the quality of the pulp which is being stored. All important. Okay. So I think I'll give you another example. What I understand nowadays is we have many banana chips. The banana chips, when you make a chip, some of they don't give a real taste of a banana. In order to improve the taste, what they do is they extract few metabolites from the banana peel, and then they mix it with the chips so that it becomes more tasteful. So this is being done in a different scale. Okay, another level in the
Alternate for antibiotic. Well, we are feeding artificial feeding of antibiotics, and we combine the bacteria with antimicrobial or antibiotic properties along with the system. Whether we can improve the antibiotic properties in the
I don't want. So far, we have not described or I haven't got any information on the microbiome associated with the honey. We know it is antibacterial, antimicrobial, and the reasons were like what are the microorganisms which are present in it, which can have we are concerned in terms of adulteration of honey, and we are using NMR method to determine the quality of honey. But microbiome tracking can help in deciding about the quality of honey. But your idea of using, improving the honey for antimicrobial treatment by adding microorganism, maybe an idea which you have to test. Somebody can pick up and test it. Right now, I have no information.
That's my research.
Oh, it's your research work. The green technology. Okay, good. That is a good idea. So you are now trying to isolate bacterium and adding to the honey?
The gut of honeybee
Okay.
constructs.
Oh, that's interesting. Are you working with Dr. M.R. Singh also?
I have, sir.
Here.
Good luck to you. I'd like to see your results. If you have been, see, honeybee, I think, if I'm not wrong, there are papers from University of Utah on honey microbiome or honeybee microbiome. So they actually honeybee, they feed on few microorganisms. I mean, the honeybee gut is having very limited number of microorganisms. Dr. S. Subramanian in IRA is also doing work on honeybee gut microbiome. So if you can list out those bacteria, use those bacteria by directly adding to the honey you are trying.
Artificial.
Artificial, yes, you can always, yes, that is an idea. That is like use of probiotics for honey. They've been doing with the lactobacillus, they've been doing with the, we have chosen the market last few years. There is a product called Yakult. It's been sold for 50 rupees, small container. It's a probiotic for human beings. That's an organism isolated by a Japanese researcher in the 1960s or 70s. It is being now sold across the world. So probiotic was anything, yes, it is being done. Good luck.
Honey is a prebiotic, sir.
Prebiotic. Look, he is trying to develop probiotic for honeybees. He's trying to isolate organism which can be taken up by the honeybee for its growth. Yes, we can. Honeybee microorganism also has got the social behavior is also related to microorganisms. Honeybee, some of the genes are turned on by the jelly, royal jelly. It is again related to honeybee microbiome, gut microbiome. It becomes epigenetic. Several interesting results are there on there. Thank you very much for your patient listening. Thank you for your invitation. Thank you, Dr. Kumada, for calling me to come and present a talk here. I mean, although I've been traveling many times this way, the campus, I never came back to this college after I left long ago. And this is my homecoming. Thank you.
Vice Chancellor, respected Dean for Great Studies, and respected Dean Agriculture College and Research Institute, Madurai, and our beloved chief guest today, and respected and beloved friends from the TNA faculty who have joined in online, and also my dear friends joined in offline. So on behalf of all, I thank you, sir. Thank you very much for your very nice lecture, and you started from life to the linking the life with microbes. So and finally, we could able to, our students could especially able to understood that no life is there without microbe. So that time, just I want to say one example of one incident, sir. So when I am doing my PG here, the next day was the Farmers' Day. So they all preparing for the stalls overnight, and soil science stall next to microbiology stall. So soil science students they have made a rangoli with they have given name that. Something the soil is very important for the health of plant. Then immediately we thought that as microbiology students, we three were there that time. And immediately we have written that no soil is there without microbe. So that likewise today the lecture went on well, sir. It's very much useful to our students. And also you have given novel innovations and ideas in the microbiome research. And so, dear friends, I want to say about Sir, because even though he's having very busy schedule now because of his student thesis submission and all other work, and he has traveled all along the way. And today he has justified his presence here. Thank you very much, sir. Department earlier, so it's our pleasure to have you today, sir. Thank you very much. And thank you, students. Thank you very much for one and all for your patience learning. And also, I thank our university and Vice Chancellor Madam and our Dean Postgraduate Studies to give this kind of opportunities to see our researchers and friends from other institutions. So this is our chance for us to see all of them, to interact with them. And today I learned somebody are working in my line in this institute. So likewise, we could able to get more information from other researchers in person when compared to talking over phone or writing letters or email. And also my students also enjoyed with your lecture, I think so, sir. Thank you very much for you, sir, again. And thank you very much for the students. Thank you.